All right. Hello, everyone. Welcome to Skype a Scientist Live. Happy DNA Day. Today is the anniversary of the day that we celebrate a couple things in DNA history. All right. Look, we got some DNA modeling ready to go already. So DNA Day celebrates the completion of the Human Genome Project. Is it ever done? No, not really, because there's always more to learn. But the initial completion of the Human Genome Project and the photography of DNA long, long ago. So today we are joined by Dr. Lawrence Brody, and we'll be getting a tour of a real lab, and then have all of your questions answered, as many as we can get to anyway, in the next 45 minutes. Housekeeping. Please submit your questions that you have to the Q&A section on the bottom in your Zoom screen. We will try to get through as many of those as we possibly can. Other things going on tomorrow, we're going to be talking about medicine that can be pulled from plants. So we'll be talking about how that works, the chemistry of acquiring new medicines for people from plant life. With that, I'm going to hand it over to you, Dr. Brody. Thanks for joining us.
Hello, everyone. I'm Larry Brody. I'm a human genetics researcher at the National Institutes of Health, which is outside of Washington, D.C., and it is at this point, the world's largest biomedical research facility. So if you're ever in the neighborhood, come and visit us. It's a government facility that you can look through. What I thought I would do is, before I sit down again, is just do a quick walkthrough to show you a lab. I'm sitting in the middle of an active research lab, which is also why I'm wearing a lab coat. And I just thought I'd show you what a real lab, as opposed, I know some of you have seen labs on TVs, which tend to be a little bit shinier and less chaotic than ours, but I'd show you what a real lab does. And so this is the kind of space that people would work in if you were working in this lab. And I'm going to just walk around briefly. And here are some people who are in training, who are going on to bigger and better things next year. And here is someone actively working, so we won't interrupt him. He's moving tubes into centrifuge. So we do a lot of spinning. There's a lot of small stuff. And I just want to show you one other thing. There's a lot of machines, and you can see it's a lot of jumble of stuff. We also have to freeze a lot of things in order to keep our samples safe. So this is just a whole room full of freezers that go down to minus 80 degrees Celsius, which is very, very cold. And in this little corner right here, where there's these cardboard boxes, it looks like my basement in some ways, are two really old automated DNA sequencers. These are the kind of sequencers that were used to sequence the human genome, but they're so old and inefficient compared to what we do now that they've just been mothballed back in the back corridor. Here I'm walking through, you can see we have plants growing, more investigators. We spend a lot of time working on computers because we generate a lot of data. So for those of you that are interested in biology, but maybe a little squeamish about squishy things like cutting things up, and you can do a lot of biology on computers. And the last thing I just want to show you is my office. That'll give you some comfort when your parents say your room's too messy. You can say, we visited Dr. Brody at the NIH and his room was a mess too. So now I'm going to get back to this corner so we can work on your questions. So I will tell you, I've been doing science for 30 years now at the NIH. And as a student, when I was your age, I really wasn't sure what a scientist did. But in college, I got to see and work for some scientists doing some research. And it looked really fun. And the thing that attracted me to the field of science was that my professors, who I thought were really ancient, I now know they were younger than I am now, seemed to be really enjoying their job after doing it for years and years and years. And I think that's because we constantly get to do new things, which really keeps it very interesting as a profession. And as I said, my specialty is human genetics. I try to figure out what is in people's DNA that might get them to be more susceptible to some diseases compared to another. And at times we try to figure out how to prevent those susceptibilities. But I'm happy to answer any questions you have. I don't know if, Sarah, you're going to moderate, or should I look at the chat?
Yes, I will be reading the questions out to you,
Okay,
so you don't have to
cool.
worry about the moving pieces here. Yeah. Before we launch into the questions, could you tell us what some of this stuff is behind you on a lab bench? Like, what are the main tools that we use when we are doing molecular biology?
So molecular biology is funny. We really do work with molecules, and molecules are invisible. So this DNA molecule is a representation of something that's really tiny and invisible. And what's pretty amazing is that we spend a lot of time working in test tubes that are about, sorry, I'm shaking them out because this is a real, are about this big. And we can do experiments in a drop of liquid that's about the size of a tear in these test tubes and learn a lot about DNA and the information that someone might have in their genome. So our genome is all the collection of all your genetic information. And so we can analyze your entire genome in a little tiny tube like this by doing certain experiments and putting in different chemicals, and sometimes we put in enzymes. You might say, how do you get it in there? So one of the tools we use, this is called a pipette man or a pipetter. And sorry, weren't prepared for the real demonstration. What I get, you take these plastic tips. See, there's a box of these tips, and you put these plastic tips on there, and then you press the plunger, and it can pick up. I said the whole volume could be the volume of a tear. You can pick up 1/10 of a tear volume and put it in there to mix things, and then these accurately dispense the amount of chemicals you need. And then when you're done, you just eject it. Usually not on the floor like that, but I just did that for fun. Sometimes we have little pipette wars where you shoot stuff at each other, or you fill them with water and shoot it at each other too.
The teeny tiniest water gun.
Yes, yes, they are very, very expensive, teeny tiny water guns. And as you saw earlier, when Dr. Wayne was putting stuff in a centrifuge, that's what these are. We do a lot of things times where you want to take the tubes and put them in a machine like this. It's hard for me to see this. And when you turn this machine on with the lid closed, it would spin, and that puts everything down in the bottom of the tube so we can work with it. Another relatively strange thing that we use all the time is this thing called a vortex genie. And believe it or not, all this thing does is shake. I don't know if you can hear that or see it. So you would take a tube, and if you wanted to mix all the chemicals, you put it on that and it shakes. It is not good for milkshakes. You wouldn't use it for milkshakes or anything else. And then the last thing that's sitting on the bench that we use pretty frequently, it's not, since this is a real lab, it's not running right now. These are called gel boxes, which you may have seen. You can put a gel in there and load the product of what you're looking at from this tube into little tiny wells on the gel and apply electricity, and it separates the DNA molecules. And I'm pretty sure some of you will, if you've not already done this, have done this in your classes. And then you can actually use a special light to see the DNA molecules. But what you're seeing is millions of them in one spot. And not to be totally cliche, we actually do use microscopes. Biologists use microscopes. So this is a microscope to look at slides. This is a microscope to look at big things. And you can probably see I can put my hand under there and look at it. So we sometimes work with zebrafish. It's a small fish that you want to look at under the microscope that way. And so that's some of the tools that we use, and people use them every day. As I've said, the computer is an incredibly important tool in biology now too. So when everyone who's out there has a desk, everyone's desk has a computer on it, because there's so much really cool data out there to analyze. Some of it you in your classes could analyze. There's a lot of it that's out there in the public.
Very
we
cool.
can probably provide some samples on how you could analyze human genomic data right in your home or right in your classroom.
Awesome. Well, thank you. That was a great tour. That was very reminds me of my time in graduate school. Okay, so we already have lots of questions coming in. So let's get to it. Our first question is from Isaiah from Locust Grove, Virginia. Can human DNA mix with other animal DNA? And if it did, what would happen?
So thanks for that question. When you say mix, I assume you mean not just put both of them in a tube, right? Because we can easily do that. We can get animal DNA and put it in a tube, and then what we have is a mixture. It doesn't do anything. But what we can do is, and what we do is we splice bits of human DNA into other organisms, usually into a bacterial, what's called a plasmid. And that allows us to analyze the human DNA very much more carefully than if it's in the mix. I don't know if in class, if you've heard, but the DNA in every single one of your cells is over 6 feet long. And it's just amazing that it actually fits in there. It's also 3 billion bases, which is really hard to think about. But if I were to type the human genome out on a piece of paper, regular paper that you use in class, and use a regular font that you would use in your computer, and I was going to type the whole genome out, it would fill pages tall enough, and I know those of you in Virginia have seen it, tall enough to go to the top of the Washington Monument. So it's 550 feet tall. So it's very hard to analyze that all at once. But if I could take a page, put it into a bacteria, let that bacteria Xerox those pages, that same one page, 1,000 times, then I could really study it very, very carefully. There are also times when we do put human genes into animals and let them become animals. Most of that we do to study the human genes, to try to figure out human disease. You may have heard that there are some studies now where they're trying to make certain animals more human-like so that we could use them for biomedical donors. So good question. And yes, we can do that, but we tend to do it very carefully. And it is almost always to try to improve either crops, food production, or biomedical research. We don't do it just to make funny organisms.
Sounds good. Thank you. Our next question is from Dior. What causes extra chromosomes, like in cases such as Down syndrome?
So so does everyone know you've got 23 pairs of chromosomes, and when the cells that formed you were eggs or sperm, when they're being formed, they go through a process called meiosis, which you may or may not have had in class yet. That involves having the chromosomes all separate. Every once in a while, a chromosome gets caught behind and it doesn't separate. So then when that goes to form a person, they get this extra chromosome. Most often, it will be chromosome 21, in part because it's smallest and it has not as much information. Most of the time that happens either in the mom or the dad, slightly more often in the dad's sperm compared to the mom's egg. We don't know exactly what causes it, but it is more frequent in the eggs of women as they get older. Other chromosomes also probably get stuck in by extra, but because they might have so much more information, that cell can't become an organism, can't become a person at the end.
That's interesting because all of the eggs in a woman exist from before she was born, right?
Correct.
So what is that about? So weird.
So it's a puzzle as to whether or not you use the eggs that might have the right number of chromosomes first.
Oh.
Or that's one hypothesis. The other model is that the thing that screens out eggs that might have 7 more chromosomes, that mechanism gets weaker as we get older.
Interesting. Very cool. All right, our next question comes from Gabriella. Can the DNA program tell you if you have a disease?
Yeah, so by the DNA program, I assume you mean the DNA sequence. So those 3 billion letters, which we can now read very easily and pretty cheaply, I might add, because of the things we've been able to do for the last 20 years. They can, in some cases, predict very strongly that you will have a disease. Sometimes they can tell you you do have a disease. And then other times, and more often, looking at the variation that's in your own genes can tell you might be slightly increased risk of disease. That's the one that is the most common and most frequent. It's also fortunate because it's the easiest one often to intervene in, because if we can predict that you have a slightly increased risk of having diabetes or having heart disease or getting cancer, some of those we can prevent. And so knowing that before you get the disease might be good. You should also know there's a lot of variation in all of your genomes. So you're a fraction of a percent different from the people in your classroom. But most of those variations don't really have any meaning at all. They're just markers. They don't mean anything. They're not going to change your biology. And then some things are really complicated, like height. So if you look around your classroom, you're not all the same height, right? And that's because there's hundreds and hundreds of genes that act all together to produce your height. And it's really a complicated thing that we can't predict exactly how tall someone is from their genome any better than by looking at their family.
Right. Very cool. Thank you. Our next question comes from Callista. How do we know what DNA looks like?
Really good question. So in the early 1950s, when this structure was determined, we had to make DNA molecules, the kind we would study in a tube like this, dry them down, make them into a crystal. So, you've probably, I'm sure some of you have spilled something and you come back 3 weeks later and now it looks like it's a crystal. They would then use X-rays to shoot through that crystal. And how the X-rays went through allowed a very famous scientist, Rosalind Franklin, to take pictures of the way the X-rays were deflected. And from that, they can back calculate with lots of math that I don't understand at all, what the structure should look like. And so Rosalind Franklin took these pictures, and Watson and Crick and Maurice Wilkins saw these pictures, and it helped them deduce what DNA looks like. If you fast forward from the 50s into the 70s, we have electron microscopes and DNA molecules. You don't actually see the DNA per se, but you can put them in an electron microscope and use a stain that allows you to see where all the linear DNA molecules are. And even more recently, we have even more powerful methods to look at the structure.
Awesome. Thank you. Our next question is coming in from Dior. What does DNA have to do with cancer?
Oh, great question. And I'm sure many of your families have been affected by cancer. And you can think of DNA as coming in two flavors in your body. The DNA that is in the cells that made you, these are the germ cells, the sperm and egg. All those cells then copy their DNA in order to make more cells and more cells, right? You only start with one cell, a fertilized egg, and that copies the DNA over and over again. Every once in a while, that DNA makes a mistake, and that mistake can lead to those cells growing too much and creating a cancer, growing in an unregulated way. Those changes in DNA in cancer are not passed on because they're not in the cells that you could pass on whenever you create the next generation. And there's a fancy term for that called somatic, which is the root for soma, which is just body. So those are your body cells. Sometimes cancers, there was a question earlier about chromosomes. They have like totally messed up chromosomes. And that also is one of the things that leads them to grow uncontrollably. The really cool thing now that we couldn't do a decade ago was we can sequence the DNA in that tumor, see how it differs from the DNA in the rest of your body. And then sometimes there are drugs that will only hit the tumor. I'm sure some of you know how awful cancer treatment can be. And this is a promising technology to make it a lot more specific and make the patients a lot less sick. They get sick from the treatment that's hitting the wrong cells. We only want to hit just the right cells. So there's a lot of effort to sequence the DNA in tumors to help treat them.
That's great. Hope that gets better as soon as possible. Great.
Yeah.
Thank you. Our next question is from Karen. How do you know, or do you know how fast a centrifuge spins?
We do. In fact, we use centrifuges that spin many hundreds times a minute. to thousands of times of minutes. And then we have a really fast one down the hall that we don't use as much anymore, that looks like a giant washing machine. And that can spin 80,000 times a minute. And that's engineering that does that. It's tested and calibrated. Same way many of you have probably been in a car and you see some of you will have a gauge in the car that says how fast is the engine spinning. There are ways to record that back from the electrical inputs. So we use different centrifuges when we need to squeeze things down in different amounts. Obviously, the thing that spins at 80,000 times, if you were in a tube and you were spun like that, you'd just be a little puddle on the bottom. So we wouldn't do that. And you're too big to go in that centrifuge anyway.
Yes, thank you. Our next question is, how do you controllably cut DNA?
Great question. There are two ways you can controllably cut DNA with enzymes. One that will just cut DNA regardless of what the sequence is, and you can do that in a controlled fashion. So those cuts are random. But then there are enzymes which cut it at very, very specific sequences. And those are called restriction enzymes. Those are the enzymes that were discovered by a group at Johns Hopkins University who won the Nobel Prize for that, because if you can cut DNA in a specific spot, you can then put the DNA molecules back together in that specific spot, like a puzzle. If you cut it randomly, you may not be able to put it back in any particular form. So it's these very specific enzymes, which, believe it or not, have evolved in bacteria to have them fight viruses that might get inside their cells. So we didn't invent these things. Nature invented these enzymes. And bacteria have these enzymes that cut up DNA. They're designed to cut up DNA that infects them to fight off it's their immune system.
Great, thank you. Our next question, what does the majority of your day in a lab consist of? For example, is it more administrative, running tests? What are those tests? Is it PCR, etc.?
So I'll give you what my day my day looks like, mostly because I am become old and senior and don't know how to do a bunch of the stuff in the lab and rely on students and really talented people to actually do the hands-on parts of the experiment. So I spend a lot of time on the computer and in my office, and then talking with some of the people you saw on the tour about how their experiments are going. As you start in the field, and as I started in the field, you learn how to work at the bench. So I started mushing up fish for a genetic study when I was an undergraduate. And then eventually you learn how to do PCR. We have PCR machines and recombined DNA into molecules and grow bacteria. You learn kind of as you go. You don't have to have all those skills before you become a scientist. You learn them as you need. I still enjoy being in the lab, but this is the fate of someone who starts to run a lab. You have more people who know more than you do, which is great. But it means you don't get to do as much as I used to.
Totally. Awesome. Thank you. Our next question is from the B9ers third grade class. Is it the same to get DNA from hair, skin, or blood? And do you ever help police identify criminals based on that?
So there's two answers to the question. One, it is the same DNA. So the DNA that is in almost all of your cells is the same. So if I get DNA from your toe or your liver, it has the same information, the same couple of billion bases of sequence. Technically, extracting it is a little bit different from those different sources, but that's a minor thing. I will tell you that we don't work in the criminal justice system. We talk to them, and we talk about technology, and we also talk about ensuring that they don't get involved in our research samples. We don't get involved in their crimes. There are a whole certification process in school to become a forensic geneticist. It's not something we do here at the NIH because we're mostly focused on health and disease research. But there's a lot of overlap in the techniques. So a lot of things that we created to do our biomedical research, the FBI and the crime labs also use. And same thing for the computer searches and the genetics.
Similar techniques, different applications.
Yeah.
Yeah. Cool. This is a fun question from Ricky from Tucson. Can one strand of DNA be all the same nucleotide, like adenine?
It can be, yes. So what Ricky in Tucson is talking about is in DNA that doesn't contain a huge amount of information because it's all the same letter over and over again. And there are stretches in our genome that have big stretches of the same letter over and over again. You can make them, so we can also make synthetic DNA, small segments. We can't make synthetic DNA the size of a person's genome or even a bacterial genome. We can't even make that. But there are those stretches. Strangely enough, there's not a lot of them because the enzymes that copy DNA, and I think you probably, for the person who asked questions, knows that it's double-stranded and one strand is used to copy the other strand. If there's a whole stretch of A's, it's hard for the enzyme to know where to stop, and sometimes those slips. So they don't stay in the genome, they mutate a little bit longer. And they don't have a lot of information. The key to the information is having lots of different letters.
Great, thanks. The next question comes from a third grade classroom in Evanston, Illinois. If we gave human DNA to an animal, would you be able to get the animal to talk?
So I think you mean if you engineer some of the human DNA into animal.
Yeah.
We are so far from being able to understand how the brain works, how talking works, all of those things. We couldn't do it.
Too complicated.
Could
What about
we?
like a smaller thing, like getting something that an animal can make or do, and then get a human to make it? Can we do that?
We, in theory, could do it. In practice, we don't because it's dangerous, right? And it's not, the time that we would manipulate a human's DNA is really reserved for the most serious of diseases when there's nothing else that would work. Because by manipulating a person's DNA, you could also mess up their DNA. So we don't want to practice on people. We don't want to experiment on people, especially on their genome, even if it's just on the DNA in their finger.
Yeah.
You have to be really, really careful when you're doing anything with people.
Very good.
So not something we do. The flip side is, yes, we do sometimes do this in animals, and there may be a way to have cows to put a human gene into cows so they produce a very valuable substance like insulin in their milk. And all you would have to do is milk the cow, purify the insulin out, and then you could give that to diabetics. So there's a lot of agricultural uses of genetics and recombinant DNA, as it's called, in that area. But even that has
I vaguely remember they put like spider silk DNA into a goat, and then they were like producing
milk.
Yeah, super cool.
Yeah.
All right. All right, we got a question, another one from Locust Grove, Virginia. Can you tell us about DNA in twins?
Twins are interesting. So remember, twins come in a couple different kind of flavors. One, which we use the term monozygotic, which mono just means one, zygotic means egg. So they came from one egg that split. Those are what we call identical twins. And their DNA is almost 100% identical. Until we could sequence the whole genomes, we would have said, yeah, it's 100% identical. Now we know there's occasionally changes that after the egg split, one twin might have one change and one might have another. Almost always those are no consequence. So they're identical. Their genomes are identical. And then what's called fraternal twins is actually when two different eggs get fertilized. And that's why fraternal twins could be male and female, whereas identical twins are always all the same sex. So fraternal twins are the same as sisters or brothers or siblings. They just happen to be born in the same time and grew up at the same time. The fact that they're similar, even though their genes are different, really tells you how important all the environmental parts are, right? You know, how they grew up in the womb and what they did afterwards. But identical twins really are very, very special genetically.
Very cool. Thanks. Our next question is Olivia from Jenkintown. How can you tell a cancerous cell from a normal cell?
So really good question from Jenkintown. I grew up in Philadelphia, so I've even been to Jenkintown. Nice. There's two ways. In the old ways, we would use, I showed you some microscopes. Remember I said cancer cells have a couple of different properties. genetically, they could have a whole bunch of extra chromosomes. They could be missing chromosomes. You could see that under the microscope. Also, tumor cells often look different from normal cells. They might be blobby, or they might be lumpy, or they might be bigger or smaller. But then the way we do it now is that we can also extract DNA from those tumors and then read the sequence of the tumor DNA. And that's the strategy that I said earlier, where we're trying to look for weaknesses in the tumor to try to attack that tumor to knock it out.
Cool. Thank you. Our next question comes from Jen Tierney. What specific gene or disease are you or your colleagues currently working on?
So that's we have a whole institute here. And
Yeah.
so there's a lot of different diseases. The lab that I'm in, they work on diabetes. Is that Francis? Okay, so and a syndrome that causes premature aging. Down the hall, they're working on cancer by looking at cancer in dogs. Down the other hallway, they're looking at why we can't regenerate hearing when we lose our hearing. So it's for you, don't listen to loud music. That's one way to lose your hearing. But for people who do lose their hearing, they're trying to figure out why we can't regenerate our hearing, because zebrafish, the fish I mentioned earlier, can. So they will sometimes expose zebrafish to loud music and then study them, and then the hearing can come back in those animals. We have 17 different senior scientists, 20 some other scientists, and I'm sitting at the National Institutes of Health, which has 1,000 different labs who are working on every aspect of health and disease, sometimes using people, sometimes using what we call model organisms. My lab is trying to understand the connection between genetic variation and nutrition, specifically some vitamins, on why some people are sick and why some people aren't.
Yeah, thank you. That's
Any
very cool.
topic you want, you can find it at the NIH.
I believe it. Our next question is, what is your favorite part of working with DNA?
The favorite part is that it connects me to biology, which I've always liked. As a little kid, I would look at ants. I've had a zillion pets, still have pets. And so to me, that's biology. And I like being outside. I like seeing all of life, grass, trees, caterpillars, worms. But I also like the constancy and the predictability. So DNA connects that biology, which is kind of maybe a little harder to predict, with the information that we can extract by looking at DNA and reading the DNA code, and then doing real predictions that are much more likely to be something that we can do at will. Or whereas if you were to watch 1,000 butterflies, which I love doing, they would be flying all over the place. Right? If I extracted DNA, I might be able to figure out exactly why their wings are certain colors. And so it's that real prediction with the real life kind of thing. So it combines chemistry, physics, computer science, and biology.
Very cool. Thank you. And I think people sometimes underestimate how complicated studying animal behavior is. It's just so messy and so challenging. It's really hard. Okay, next question is also from Jenkintown. We got Chloe from Jenkintown. Can you cut the disease part of DNA out?
Great question, and something that we have been working. This is the royal we have been working on for a very long time. So if you've inherited a part that's going to make you sick, can't we either get rid of it or just swap it out for a good part? And it's really hard, in part because we have to get it to the right cells. So imagine what you have is a variation that affects your liver. Getting the new DNA to replace it in your liver, which has a couple billion cells, is hard. The thing that we have now able to do, and some of you may have heard of CRISPR, is a method that allows us to cut and paste and swap in the DNA much, much easier than ever before. It's an amazing advance that's happened only in the last 10 years. That doesn't solve getting it in the right place, but there are some promising, this whole field is called gene therapy. And sometimes it's taking out a bad gene, sometimes it's putting back in a good gene. And there's some really promising advances now. One of the most promising is in sickle cell anemia, which some of you have probably heard of. It's where your blood cells are funny shapes and they cause pain and really lots of distress. There's a therapy where they manipulate the cells that are going to make the red cells to make a slightly different kind of hemoglobin so that they don't get that weird shape and clog up your arteries. And that uses this CRISPR-Cas. So I think for you students, you're going to see many more advances in this area than I've seen over the past 30 years. I will tell you, it's been a struggle for 30 years, not me personally, but watching the field have a very, very difficult time. Do what should be easy. Take out the bad one, put in a good one.
Yeah, easier said than done.
Sounds easy, yeah.
Yeah. Here's a pop culture question. Bridge wants to know, our class wants to know if you know that Lizzo song, I just took a DNA test. Turns out I'm 100% that scientist. And is it popular among scientists?
I have heard it. Yes, scientists probably are a little out of tune with pop culture at some point, but I have heard that song. I can't tell you if I know what it means, but I like the song. I like the song.
Good song, good song. Karen wants to know, do you work with anything explosive?
We actually do, but not something that we hope to explode. So we often work with ethanol, alcohol.
Oh, yeah.
And you have to be very careful with that. We sometimes work with ether, which is another compound that can be explosive, but I didn't go to it. But for those of you, many of you will have chemistry at some point, and you will do things in what's known as a hood, which is a way to make sure that all the vapors go up and out and don't harm you. The explosive stuff is also not good for you. So we really try to minimize that. And so I have flooded a lab by accident once, but I've never blown anything up by accident. And we try to.
Was that zebrafish related?
No, it was no, it was a plumbing accident. I was putting speakers up in this lab right here, and I punctured a pipe by accident.
Nice. I have also flooded a lab, but it was squid related. So aquariums, you're just gonna flood stuff sometimes. Okie dokie. Then here's a question from an anonymous attendee. Oh, Ashton wants to know, what's the most dangerous experiment you've ever participated in?
One of the most dangerous things we do, believe it or not, is, so I'm not a participant, but is a laboratory method. So those gels I talked about, we are poured out of a seaweed gel. So if you can think of it as a special kind of jello. And in order to get it to be liquid, we heat it up in the microwave. And we heat it. I don't know if you've ever heated something in the microwave that gets superheated so that as soon as you touch it, it starts to boil. Strangely enough, that's one of the more dangerous things we do. I always tell people to be very, very careful with that because it's hot, sticky liquid. And the last thing you want on you, and we do wear gloves and lab coats as much as possible, but you don't want hot, sticky liquids on you. The other thing we occasionally do, less so now, is we would sterilize things with a Bunsen burner and a little alcohol. Sometimes that spills. But in reality, we don't deal with hazardous stuff very, very often. And in my career as a scientist, it's decreased the amount of hazardous stuff that we deal with.
For sure, yeah. Our next question is from Charlotte from Jenkintown. How did we discover that zebrafish are so good at being model organisms?
There was some pioneering work done in Germany and in Boston by a series of investigators who wanted to look for something that is a vertebrate. So if you think about the models we study mice. Sometimes we study, not me, but some study is done in primates. They don't have a lot of offspring to study. And then we study things like fruit fly or nematodes, these worms, or yeast. They have lots of offspring in short generation times, but they're not vertebrates. And so the nice spot in the middle is zebrafish, which is a very small vertebrate. So it's really very, very similar to you compared to a fruit fly, even though you might not think of it that way. It's very close to you in evolution compared to a fruit fly or a yeast or a worm. And they can have hundreds and hundreds of offspring. So these groups started saying, hey, we should study these. And it just kind of spread like wildfire. The other major advantage is that after you fertilize the sperm and the egg in the zebrafish, the first 4 or 5 days of development, you can watch because it happens in a ditch. And if you look around on the web, you will find movies of a zebrafish going from a single cell to a fish. And you can watch that all happen in a 3 or 4 day period. So
Super.
lots of offspring, they don't take up a lot of space. And they're vertebrates, so they have almost all the same genes we do. Different flavors of them, but almost all the same genes we do. And
Awesome.
the stuff we can't do, like breathe underwater.
That helps. Yeah. All right. So we are almost at time, but I wanted to get one more audience question in. There have been a number of people asking the question, like, how do you get this DNA? Where does it come from? And I'm wondering if maybe they're asking that because they may have heard of like Henrietta Lacks and maybe read The Immortal Life of Henrietta Lacks. Could you talk a little bit about like how samples come to be and how that's changed over time?
Sure. So it's not how you pull DNA out of cells. The question
How we
is,
get the stuff
yeah.
that we then get the DNA out of. Yeah.
So, and you're right. In the time of Henrietta Lacks, we didn't have systems to do this, and people thought I could just use this without asking anybody. We have moved forward a lot from then in that we now have, if we're talking humans, right?
Right.
But the zebrafish, we don't actually ask
Ask them. Yeah.
but in humans, there's a process that is called informed consent. So everyone who participates in the study is a participant. They're a partner in the study. They're told what's going to happen in the study. They're told what's going to happen with their DNA. They're told why the study is going on. And they have the ability to say, okay, I'm out and tap out of the study at any time. So there's a whole bunch of guidelines in order to protect human subjects, is what it's called, even though we now call them participants, not subjects, or partners in research. And so no one's DNA should be used without their permission going forward. In the Henrietta Lacks time, we didn't have those things in place. And so lots of people, especially people whose samples were taken as part of medicine, were either going to get thrown away or used for research. So they thought it was the better of the two options. But if you students were in a study, you would actually be asked. Even your parents cannot make you be in a study if you don't agree. There's something, even though you're too young to legally sign into a study, you would be asked for your assent, and you would sit down with someone and say, you're going to be in the study. This was about, do you agree to be in the study? Do you understand it? And you agree to be in the study? So right now it's really a better situation where we're partners, and obviously families and groups of people affected with disease are very anxious to get research done. So they want to be partners because they don't want to live with these diseases, or don't want to have the next generation to live with these diseases.
Absolutely.
So that was the angle you were looking for.
That's exactly the answer. Yeah. Covering that was perfect. Thank you. Okay, so now we're almost at time. So we always ask all of our scientists the same two questions at the end of each session. The first question is, if you had everyone's attention in the whole world and you could tell them one thing about your area of expertise, what would it be?
That's a good, so if I'm a scientist, my one thing would be to understand how science works and why it's so important. As a geneticist, it would be, think about genetics. Don't think about it as destiny. Think about it as learning about yourself or about disease process, but it's not usually, in most cases, 100% predictive. So if someone says you have the gene for type 2 diabetes, that doesn't necessarily mean you're going to get diabetes, or you have this gene for cancer. So that's two-part answer. I think science is a really important process. I think it's misunderstood. I'm hoping all of you listening can understand how science is a good way to get to answers that work, as opposed to just opinion or making it up.
Totally. Awesome. Thanks. Your next question, you still have everybody's attention in the world in this hypothetical scenario, and you can tell them one thing about anything. It can be, about your favorite sports team. It can be something significant or important or silly, anything you want. What do you want? One piece of information the whole world to know.
So most of you are in the spring season, right? So go out and just look at what happens in the world when things start growing again, or there's this rebirth in biology. Look at a blade of grass and figure out how amazing it is. And look at the plant behind Sarah and figure out, how does it know to do that? And so have a sense of wonder about this stuff, because it is really cool and we don't understand a lot of it. We're going to understand a lot more through the some of you that will become scientists. I think all of you should understand science. Not all of you have to become scientists, but we'll understand more. But really go outside, look at the natural world, and just see how amazing it is.
That's a great answer. Awesome. Thank you. Thank you so much for taking the time to share your knowledge with us, to share your lab with us. This has been amazing. Is there anything else you'd like to say or plug or anything like that before we wrap?
Nope. Yes. Happy DNA Day. We have a lot of
Oh, happy
resources that if you go to genome.gov, you can find educational resources and information about lots of different topics on our website.
Awesome. Thank you. All right. So that's it for us today. Everybody, we will see you tomorrow talking about how scientists get new medicines for people from plants. Really underlying an enjoyment of outdoor animals and plants and everything tomorrow, mostly talking chemistry tomorrow. Otherwise, we still have so many scientists available to get matched with classrooms. All of our scientists are volunteers. They're ready to go. You can talk about everything from like penguins to outer space. Sign up and tell teachers, tell your colleagues that we exist, because we're free and we've got lots of scientists that are waiting to be matched with a classroom like yours. So feel free to sign up. Tell your friends at skypeascientist.com. Thank you all for joining us. Thank you, Jasmine, for interpreting. Grateful for you. And thank you, Dr. Brody.
Thank you.
Bye. Bye, everybody.
Bye-bye.